Well, good evening, everyone, and a very warm welcome to our new series of COVID Connections discussion events, which are brought to you by Welcome Connecting Science and the COVID-19 Genomics UK Consortium, or called UK for short. Over the next few months, we'll be exploring the coronavirus pandemic through the lens of genomics and how understanding the DNA of the virus is so vital in how we're not only keeping one step ahead of it, but indeed also creating better health outcomes for people. So we'll be looking at this through a range of different perspectives. All our speakers over these events have connections to the UK-wide genomics effort, the COG UK effort, which comprises a mix of universities, public labs, NHS centres, research campuses, including big efforts going on here at the Welcome Genome Campus, where we are hosting, at least virtually, tonight's event from. We're very keen on these events being about conversation. We want to hear not just from the people involved in the science effort itself, but also from all of you who may have questions. You may have indeed experiences around the virus, views to share about the pandemic, and in particular, how understanding the virus at the DNA level is helping to make a difference. So we'll be leaving lots of time for questions and discussion. And if you do have a question or a comment, then please use the Q&A feature on the Zoom page. Now you'll either find that at the top or the bottom, depending of your screen, depending on the device that you're using. We always say this at these events, but please don't worry if you think you're the only person asking a question. It's everyone sees that view, and it's because they come in a feed to us, and we look for themes that we can maybe cluster so that we can get as many perspectives to the panel as we can in the short time that we have. So please do type your questions or comments in that Q&A box as we go. We've also got the option for closed captions this evening, and so to turn those on, please look for and then click on the live transcript button, which is next to the Q&A box, and then choose the option Show Subtitle. So, so far tonight, I'm delighted we're joined by 120 people, and there's more joining as we speak. So it's really great to welcome you all to the event. So first, I'd like to just introduce this evening's speakers and before handing over to the chair for tonight. So firstly, I'm delighted to welcome Dr. Catherine Bladden, who's Director of Operations for the COVID-19 Genomics UK Consortium, and is responsible for the long-term operational strategy, coordinating public health investigations, and engaging with a wide range of collaborators that are involved in the genomic surveillance effort. Catherine is a genomic epidemiologist with expertise in pathogenomics, in antibiotic resistance, and in clinical microbiology. Prior to joining COG UK, Catherine was working with the European Centre for Disease Prevention and Control, where she supported international outbreak investigations using whole genome sequencing datasets. She's a One Health expert, which is about bringing different sectors together for better health outcomes, and was previously a Sir Henry Wellcome postdoctoral fellow at the London School of Hygiene and Tropical Medicine, where she actually used a One Health approach to understand transmission of antimicrobial resistant E. coli. Also joining us on tonight's panel is Verity Hill. Verity is a PhD researcher based at the University of Edinburgh. Before the pandemic, Verity was developing new techniques to combine genomic and non-genomic data from the West African Ebola epidemic. And that was about creating new insights that could end future outbreaks more quickly. Now she performs large-scale analysis of the coronavirus genome, shedding light on how the virus got to and spreads within the UK and how it's evolving. This includes studying mutations, for instance, the well-publicised Kent variant that was detected, as many of you might remember, in late 2020. Verity's work also helps researchers across the world track and investigate the virus, so it has a real international impact too. And then thirdly, on the panel tonight, we've got, I'm delighted to welcome Dr. Jeffrey Barrett. Jeff is director of the Wellcome Sanger Institute's COVID-19 Genomics Initiative. And that sees samples from positive tests from around the UK sequenced and analysed in near real time now in order to help guide public health response to, in particular, to local outbreaks. Prior to this, Jeff studied the genetic basis of human diseases in both industry and academic settings. Most recently, he was Chief Scientific Officer at Genomics PLC, and before that, Director of Open Targets. Open Targets is a public-private initiative that uses human genetics to improve early-stage drug discovery. He was group leader at Sanger for 10 years, where he focused on the genetics of inflammatory and neurodevelopmental diseases, and is fellow of the Academy of Medical Sciences. So welcome to our panel, and also welcome to tonight's chair. So I'm delighted to welcome Philip Ball. Philip is a science writer and former editor of Nature, where he worked for over 20 years, first as an editor of physical sciences, and then as a consultant editor. Phil's the author of many popular books on science and has contributed to discussions on science, culture, and society on many occasions in both radio and television. This includes presenting science stories on BBC Radio 4. Phil's many books offer fascinating perspectives on science, on human behaviour and discoveries, and the relationship to broader culture and society. And this is well illustrated in his latest books, How to Grow a Human and the Modern Myths. So, without further delay, I'm now delighted to invite Philip and the panel to the Zoom stage for tonight's event, which is called Shedding Light on the Invisible, How Genomics is Helping Defeat a Pandemic.
Thank you very much, Ken. Okay, it's an act of faith always that everyone can hear me, but I'm going to take it for granted that you can. Thank you for joining us this evening. Ken has set the scene for what we're going to discuss. I want to just say a few words more about that scene. I guess I felt I wanted to begin by saying that I think you'd find it hard to find anywhere else in the world that has had such a contrasting combination of success and failure in the response to the COVID pandemic, as we've seen in the UK. So the failures and their consequences are all too apparent in the high death rate, the severity of the economic damage, and I think the late decisions that have led to that. But I think we should recognise the successes too. The work done at Oxford in the development of a cheap and safe and reliable vaccine has obviously been one of those, as has the handling of the vaccination programme in the UK itself. But less widely recognised, and yet arguably just as vital to the response, has been the work done on tracking the coronavirus by genome sequencing, and in particular by COG-UK. And COG-UK began from scratch right at the outset of the pandemic, back in January of last year. It was formally established in April, and it includes the UK's 4 public health agencies and many academic institutions, including the Wellcome Sanger Institute and the Lighthouse Labs that were created for analysis of the COVID tests. As of mid-January of this year, there will be obviously more up-to-date statistics now, but as of this past January, COG-UK had sequenced more than 200,000 coronavirus genomes. That's around 10% of the total number of COVID-positive tests. And those results provided vital information on how the virus is spreading, where it's coming from, and now in particular about the appearance of the new and potentially more dangerous variants. So when we hear about, for example, how prevalent the South African or the Indian variants of the virus are in the UK, this information is primarily being gathered by genome analysis. And what's more, it's information that's crucial to understanding if and how these new variants might evade or reduce the effectiveness of the vaccines and how to respond to that with new vaccine formulations. The genome tells us what specific mutations are in the virus that make that difference. And tonight we're going to hear about this work from several experts who have been involved in it, all the way from the basic sequencing processes to the population scale data and its implications. And it's easy to forget that this isn't information that could have easily or quickly been gathered if, say, the SARS epidemic of almost 2 decades ago had become as global and as lethal as COVID has. It wasn't until the early 1990s that the genome sequences, even of the smallpox viruses, were known. And the use of genomics to track the spread of viral infections really only started to take off in this millennium. So it's really in the wake of the foot and mouth epidemic in the early 2000s. The value of genomics for the COVID pandemic is, I think, both an illustration of and a vindication of how the technology has developed over that time, over the past couple of decades. And it shows how vital both basic science and the capacity to scale it up for wide-scale deployment, how vital that's going to be in dealing with what seems to be a future world of likely future potential pandemics. No one knows how the pandemic, the COVID pandemic, is going to continue to unfold from here, but the latest modelling studies, although cautiously optimistic about what will happen as the UK opens up, they do predict a small third wave, and they suggest that the new variants are perhaps the crucial unknown factor in how things are going to go. So viral genomics are going to remain at the absolute centre of efforts to keep COVID under control. So there couldn't be a better time, really, to hear about how that works and what it can do. And that's what tonight's panelists will explain and explore. So I'm going to turn now to the first of them. We're going to hear first of all from Catherine Lodden of the London School of Hygiene and Tropical Medicine. So Catherine, over to you.
Thank you, Philip, and a really good introduction. And I think it's fair to say that everyone who's part of COG UK is very proud of the developments over the last year. And as you've explained, COG UK, which stands for the COVID-19 Genomics UK Consortium, is really a legacy that we've built from scratch, and I think the data is now being used worldwide. We have samples that are being sent to 16 sequencing sites across the UK, as described, the Central Sequencing Hub in Sanger, the 4 public health agencies, and really we use that data to detect the mutations and the variants that, as you've just talked about there, but also to combine the viral genome data with other human genomes to look at that viral human interaction. We support national and international collaborations, and we also involved in various key studies such as reinfections, care home dynamics. So that this data is used for real-time surveillance, but also to look at key research questions, and to also track and support outbreak investigations. So they're just key areas of what we use the data for. But one thing I think that I'd like to explain to everybody listening today is the huge amount of work that's gone on behind the scenes. So I'm the Director of Operations for COG UK, and essentially I'm responsible for the end-to-end from once the sample tests positive for SARS-CoV-2, that it gets sequenced on the other end. And I'm very proud to say that actually we're receiving samples from over 120 diagnostic testing labs in the UK and over 90 in England. So for those labs who have been inundated with tests and shortage of staffing and reagents, for them to really come on board and send all those samples for sequencing is a huge thank you to everybody in those diagnostic testing laboratories in the NHS and the Pillar 2, but also to the couriers and the people in the sequencing labs who then receive the samples on the other end. So just to say that that's a huge milestone that we've hit right now. And we're also sequencing samples from, as I mentioned, from the ONS survey, vaccine studies, prisons, care homes, and all those samples are being tested and then sent for sequencing. So really that end-to-end process involves so many people. So really you couldn't even list them. There's hundreds of people, and we've got Jeff and Verity on the call who've been instrumental in certain aspects, which we'll all describe. But really my voice today is just to say that this is a huge operation, from not just the sequencing, but all the people who are doing the testing, sending the samples, gathering the data, sequencing it, then analyzing it, feeding it back to the public health agencies, and then using it for clinical information. And just to say that I think it's something that the UK should be very proud of. And for me, I'm very proud to be director of operations of such a great organization. Thank you, Philip.
Okay, thanks very much, Catherine. And I just want to remind the audience, please do feel free. I see it's starting to happen already. Please do feel free to post questions in the Q&A at any time during this, because we want to pick up on them once each of the panelists has had their short introduction. Next is Verity Hill from the University of Edinburgh. Verity, over to you.
Thanks, Philip. Yeah, so as Ken said earlier, I'm a PhD researcher at the University of Edinburgh, and I am a computational biologist and a genomic epidemiologist. And I used to work on the biggest genomic data set sequenced during an epidemic that we had before the COVID pandemic, which had about 1,600 genomes in it that were sequenced during the epidemic. And at the time, that was a feat in of itself. There's now well over, I think we're nearly at 1.5 million genomes sequenced worldwide for SARS-CoV-2. So to just show like the incredible effort that's been done in the UK, but also by countries everywhere. We've got, I think there's now over 180 countries sequencing SARS-CoV-2. And these are countries that may not have been sequencing routinely before. They were maybe, they had labs set up for other diseases that they have now turned into SARS-CoV-2 labs. And it's just been a truly incredible global effort to see. And it's nice to see, yeah, that the culmination of decades of work and infrastructure building from people everywhere have come together in something that has produced this absolutely amazing data set. But yeah, so what I used to do is I basically made a giant simulator so that I could rerun the Ebola epidemic and try and work out what data would be useful in real time, so that we could work out for future epidemics who to sample when and who to sequence when, and that would give us the information we needed to work out how something was spreading. Because that's what genomic epidemiology is. It's using the information within the genome sequence of the virus, or other pathogens as well, but I work on viruses, to show how we can, to understand how the virus spreads in space and time. So that's what genomic epidemiology really is, like at its heart. And I think that's kind of mostly what we will do here. So then the pandemic hit and I was employed by COG for about 6 months, put the PhD on hold. And to start with, I was working on generating reports that would go back to COG so that we could kind of keep an eye on what was going on internally. But that kind of then went into some tools that I and other members of the Rambo Lab in Edinburgh have been developing. Specifically one tool that we're quite proud of, which is called Civit, which was a tool to help people in kind of regional centers investigate their outbreaks, so they could get a sequence off their sequencing machine straight away, put it into Civit, and that would give them the context of where their sequence fit into the wider pandemic. So that can be very useful for working out whether a hospital outbreak is really an outbreak or whether it's lots of different introductions from the community, for example. That's one of the uses it's been useful quite a lot in Edinburgh. So that's kind of one of the big projects we've done. And then now I've kind of moved on to doing tracking the evolution of SARS-CoV-2. So we did some work last year on the spike mutation, the D614G mutation, and trying to work out how that was evolving in the UK and whether it really was transmitting any faster than, we call it the wild type, the kind of base virus. And then now we're kind of now thinking about, we call it B.1.1.7, which is this lineage, this variant of concern that was first sampled in Kent. And we're asking questions about how did it evolve? Because it has a lot of mutations, like way more than you would normally expect for the time allowed for the virus to mutate. And so how did it get there? What selection pressures was it being were acting on it? And then also, how did it spread across the UK? So we've got this, as Catherine was saying, this amazing data set. And not only do we have all these genomes, they will have so much rich data with them that's connected to the genome, which it's hard to express how unusual that is and how brilliant it is, and how much easier it makes our lives on the analysis end. It means we can really get like every every like drop of information out of the genome that we can use to help people. So it's not, there's no wasted data, or far less than there would normally be. So that's kind of what we're doing at the moment. So these kind of big end of pipeline questions are what we do.
Great, thanks very much, Verity. So finally, before we get into the general discussion, is Jeff Barrett of the Sanger Institute. Jeff.
Thanks, Philip. So yes, I work at Sanger Institute, which is just outside of Cambridge, and Sanger is the biggest genome sequencing facility in the UK. It was the UK's kind of main contributor to the Human Genome Project, which was the principal reason it was founded 25 years ago. And ever since, it's been a large producer of genomic data. And at the beginning of the pandemic, when COG UK was being put together, Sanger joined as one of the partners to contribute some of that sequencing capacity. And I think what we've worked on, as Catherine really nicely laid out, there are all of these different contributors and components to the overall success of COG UK. And what we tried to do at Sanger was figure out the best way to plug in, what is essentially a giant sequencing factory into this network. And we did various things early on, and where we kind of settled was actually to build a specific partnership with the so-called Lighthouse Labs. And these are the kind of giant PCR testing facilities that were set up by the government last year and continue to operate, where most of the home swab tests, or if you go to a car park and get a test, they all get sort of centralized at these handful of places around the country. The PCR test is done, and then actually we ship the kind of waste material, the residue from those tests to Sanger and sequence as many of them as we can. And in fact, we've kept sort of building up our capacity to the point where at the moment we can attempt to sequence essentially any test that comes back with enough virus in it. Sometimes as you get tested, if you're very early or very late in your infection, you won't have much virus in the test, and it's hard to sequence the genome in that case. But for those that have enough material, we can sequence almost all of them, and that's close to half of the positive tests every day now in the UK. So we have this, and this gets then sort of immediately shared with the wider COG academic community and the public health agencies, and it provides a really fine scale view of what's happening and which variants are arising and how they're moving around from place to place in the country. And as Verity alluded to, one of the ways we've that's turned out to be useful, which wasn't maybe the first thing we expected, but turned out to be very important, is as these new variants of concern that have different biological properties, for example, they might be more transmissible than the original virus in 2020, and we don't really have very strong examples of this yet, but there are some that are a bit worrying. They might be less well neutralized by vaccines. Seeing those, trying to annotate which ones we think are worrying, watching where they move in the UK, is something that are all kind of benefits of having this data set at scale. And so right now we kind of continue to try to operate that as much as we can and as fast as we can to put those data into the hands of people who can then try to understand what the virus is doing and make recommendations on how we might react.
Great. Okay, thanks so much, Jeff. All kinds of questions were coming up for me as all of you were speaking, so I want to kick off with some of those. And I think one of the things that I'm intrigued about is, Verity, you mentioned that previously having 2,000 viral genomes was considered a lot. So it is astonishing to me how quickly this has all happened at such a large scale. I wonder if you can all talk about a little bit about how new really is this capability that we have. What, for example, has been the role previously of genomics in trying to keep track of, has it been used for flu epidemics, for example? Is this something that could have been even contemplated 5 years ago?
I can go first.
Yeah,
sorry.
No, you go first, Verity.
So so one of the things that I think is, I can't speak so much to the UK. I think Catherine and Jeff will be much more qualified to talk about that. But certainly globally, one of the things that we've been trying to push for is it's this expansion of instead of trying to do things where when an epidemic happens, people from, say, Europe and North America parachute in somewhere, do research, and then leave again, and don't build any kind of capacity in the country. There's been a big pushback against that in more recent years, quite rightfully. So there's been, over the last kind of few years, I think there's been a really big push to try and build proper long-lasting capacity in lots of different countries across, mostly Africa is more I know about, but I know that these things are happening in other parts of the global south as well. So from a kind of global point of view, the capacity has only been growing and it will only continue to grow. Well, hopefully if the UK government cuts don't kill it too much, but kind of on an anecdotal level, there's things like there was the grant network that my supervisor is part of. We went and we ran a course in Ghana for scientists across Africa to come and learn how to sequence, do basic bioinformatics and phylogenetics. And we have since collaborated with a number of those people on producing SARS-CoV-2 genomes. So there are some researchers in Rwanda who were at that course who have recently submitted a paper ready for publication, and a researcher in Botswana as well, who's also, so these kind of like direct impacts of this big effort to get away from parachuting in and leaving and actually proper training, I think has certainly been beneficial globally.
And is there an international consortium that gathers all of the global data together?
So there is a database called GISAID, which was originally set up for influenza virus sequences. What's really good about GISAID and why I think it's ended up being used is that it has, it's not completely open data. When you sign up to it, you have to sign a user agreement that you will do things like contact the data producers when you use their sequences. So it provides a level of protection for the data producers. So it's quite popular with people in lower middle income countries where historically data from those countries has often just been exploited by researchers in North America and Europe. So if it was totally free, there would be less incentive for people to put their sequences up. So GISAID is good because it has this protective thing about it. And I think that's partly why it's been so popular for SARS-CoV-2, but that's where everybody puts their sequences. So it's not a sort of organization that's deliberately collating it, I don't think, but it is a huge resource. That's where the 1.5 million genomes live.
Mm-hmm. Okay, thanks. I felt like I really wanted to get this question in before I forget. I imagine it's in a lot of people's minds, and you are the folks to ask about this. What are the variants that we should be most worried about at the moment?
I can take a stab at that one. I think there are two main properties of a variant of the virus that have got the most attention about whether we should worry or not. And those are, can it spread more quickly from person to person? And do the vaccines work against it? There's also, there's a third important one, which is how severe is the disease that the variant causes, but that one's actually just very hard to measure. So I think it's sort of doesn't get as much attention. In terms of the vaccines not working, there's a variant so-called B1351, which was the one first seen in South Africa, which I think people have the most concern about. It's interesting, there were a bunch of laboratory experiments that consistently showed that, for example, blood serum from people who'd had a vaccine didn't neutralize the B1351 virus as well as others. And that might mean that the vaccines are a bit less effective at actually preventing infection. What we've seen in some really great and reassuring real world data, because this real world data about people are exposed to the variants, they're vaccinated, do they get sick? That's the ultimate most important kind of data, more than any lab. The lab experiments are valuable, but don't sort of prove it. And in the real world data in Israel and Qatar are the two that have the most data. And they show exactly that the vaccines are a bit less effective against B1351, but not that much. And that's the absolutely critical thing. There has not been any variant yet which is just fundamentally blast straight through the vaccines. It's not impossible that that will happen, but I think there's so far reassuring information about that. On the side of transmissibility, there is one variant that has, in 2021 at least, clearly been the most concerning, and that's B117, the the so-called Kent variant that Verity alluded to, which interestingly, as of like a few weeks ago, 99% of every new case in the UK was this variant. It had just completely taken over. And in fact, we saw the same pattern happening in country after country in Europe, in North America, in other parts of the world. It just, once this variant arrived someplace, it tended to spread very, very fast. I would say the one which is now on people's mind and for which the book is still very much open, though, is the so-called B16172. These numbers get to be something of a mouthful, which is the one that was first seen in India and has spread apparently quite a lot there, although for kind of totally understandable reasons, there hasn't been much genomic data from India in the last month or so, because obviously the system is straining to deliver basic healthcare. We have seen a lot of it in the UK. It does seem to be growing pretty fast. I think as people may have seen, Public Health England designated it a so-called variant of concern alongside South Africa and these other ones a week or two ago. And that was primarily on this transmissibility evidence. And so I think we have to watch and see how fast will it grow? Will it possibly grow even faster than the Kent variant? I don't think we know yet. And I know there are a lot of people working really hard to get the information on the vaccine efficacy question, because ultimately, for a country like the UK, at least, that's the most important thing, which is we have a plan, which is to vaccinate fast everybody. And if the new variants are neutralized by the vaccines, then they don't matter that much. If any of them are less well neutralized, they obviously can become a little bit more worrying. Mm-hmm. I don't know if Catherine or Verity want to add anything to that.
I think you've summarized it really well there, Jeff. I think they are the two main characteristics that are used to identify variants of concern. So I think you can leave it open to any questions from the Q&A, but I think you've summarized it very well. Thanks.
Could I follow up on that point by asking, how does the emergence of new variants play against the question of the vaccination programs, the status of the vaccination programs generally? Because I've heard it said certainly that the worst scenario in a sense is that you still have widespread infection, but you have a significant proportion of the population vaccinated, either globally or nationally. That creates a selective pressure for vaccine-resistant variants to emerge. Is that the case? And is that something we should be concerned about, in terms of trying to get as many people nationally and globally vaccinated as we can?
I think, so the answer to your last question is clearly yes. We have to get global vaccine equity. And I think the very richest, fastest vaccinating countries it's well past time that the UK and the US starts shipping vaccines to other places because they will do more good for everyone. And that is the kind of key thing for the world, is how do we get the vaccine coverage to catch up everywhere as fast as we can. I have heard this hypothesis that partially people with one dose of vaccine create a selective environment for variants. That's possible, but I don't think there's any really strong evidence. I don't know if Catherine or Verity have a different view on this. The places where we've seen so far all those variants we just talked about, there's no evidence that vaccination had anything to do with their arising. I think the most worrying, the thing that generates new variants is transmissions, more cases of the virus. If the virus is around, it has time to mutate, and those mutations can spread. That's, I think, the thing that we would like to reduce as much as possible.
I second what Jeff was saying. I think the most important thing that we can promote is the vaccination. We know that it works to reduce the cases. We've seen it here. We've seen it in other countries. So I strongly say that I think that's the best thing that we can do right now. And in terms of the hypothesis that Phil, that you suggested about, what happens after the first dose, I think that, there isn't strong evidence to suggest that from what I've seen. And I would think that a lot more work is needed to be done to actually understand really if that is the case. And there's a huge amount of work being done to look at reinfections and look at people after their first dose and see if there is, following case. But I think that would take a lot of time to actually wait for it to do the analysis that would be powered to address that question.
Okay. Yeah, and I would just say as well that so two of the variants that we know about and are sort of sometimes thinking about, the one first found in South Africa and the one first found in Brazil, it seems that they both arose in areas of very, very high transmission and quite dense populations. So we're not sure exactly how these arose. This is part of the problem with this pandemic, the selection, we call it the selection kind of landscape. The fitness landscape of the virus is really, really complicated, it seems. We have a lot of questions to ask about why the variants all seem to be arising at once, and what impact does population immunity have on that, and what impact do varying levels of lockdown have on that, because you don't have people mixing freely, you have them kind of chopped up in small populations. And we have a lot of discussions about this, and we haven't yet come to a good conclusion about what is driving variant selection. So basically it's really complicated. And like, yeah, definitely if we can reduce transmission through pharmaceutical and non-pharmaceutical means, that's going to reduce the opportunity for the virus to mutate.
Barrett, do you know if there's information about specifically where B1351 likely arose? Because in P1 it was in Manaus in the Amazonas state, and in B117 it was in Kent.
Yeah, well, we know where it was first sampled at least, which was in Nelson Mandela Bay, which is quite a densely populated area, and I think quite a poor area as well. I also don't know about the impact of HIV positivity, because I know like some parts of South Africa have very high HIV positivity rates in adults, and differing levels of treatment availability. So I think I know less about the South African one than the one first found in South Africa than P.1, but yeah, they're probably a sort of densely populated, low socioeconomic status area with some level of immunocompromised adults, I think.
As well as telling you, as well as revealing new variants and sort of revealing where they are, I guess one thing that the genomic data can tell us about is the basic biology of what they're like. So, you know, presumably within with the mutations are showing you which proteins have changed and how, and perhaps telling you something about, you know, how that's affecting the different properties that these variants have. So I wonder if any of you can say anything about that, what we've learned about the biology of what is causing these variants to be better spreaders, for example, and whether that information can be used to, or has been used to redesign vaccines to be, you know, more effective against new variants.
Either of you want to take a stab first?
My structural biology is really rubbish. I know that the D614G mutation makes the spike protein kind of raise its arms a bit, which makes it easier for it to get into the cell. But that's, yeah, that's basically it. I think it's mostly, I know the ones we're mostly worried about are in that spike protein, which is how the virus interacts with the human cell. And that's also what the vaccine is targeting. So it's like the dual concern of transmissibility and vaccine evasion. But yeah, no, my basic biology is.
And similar to Barrett in terms of the mutations I know that have been associated with concern would be the E484K mutation that has been linked with perhaps vaccine escape, and it's been investigated. And again, there's lab work being done on those strains that have those variants to see, Phil, what you were talking about vaccines about, you know, is there evidence to suggest that there's a biological reason that these mutations could affect vaccines? So there is certainly a list of mutations and areas of the proteins that are changing that may be linking to them having more chance of vaccine escape or transmissibility. And those list of biological changes in the protein and different mutations are being monitored by the public health agencies. And so any new variant of concern, we check that list and see if there is that kind of red list of mutations, if they're in those new variants, which is as Jess said, will be linked with either transmissibility or perhaps with vaccine escape. Jeff, did you want to comment further on those?
I think those are great summaries. The only thing I was going to add was, as Catherine was saying, that, you know, that we kind of have this list now of both specific mutations, like the most famous one is probably the C484K, but also kind of neighborhoods in the viral genome where it's not exactly the same mutation perhaps, but it's the same trick the virus seems to be playing. And so, for example, the E484K is in the receptor binding motif, which is like the really the exact place where that spike protein that's dotted all over the surface of the virus sticks to a human cell. Then there's another one that's, nearby in the spike protein, the N501Y mutation that seems to be, we don't know biologically quite why, but it certainly seems to be about transmissibility rather than immunity. And then there's also things like there's this so-called furin cleavage site, which is basically that spike protein is in 2 bits. And when the virus really wants to get into human cells, one thing it does, my structural biology is exactly as sophisticated as Verdi's. I'm using my hands to show you what I mean. It splits the 2 bits apart, and actually the virus hijacks, as viruses do, another human protein to chop the 2 bits. And so there's a mutation that makes that chopping different, better, easier somehow. And then there's also, those ones, I think people who are experts have some sense of biologically what's going on there. There's also some, like, we keep seeing small deletions of like 6 bases or so of the virus.
I was going to bring those up as well.
Yeah, and it's like those ones, I don't know if you guys know, but these are in like the so-called N-terminal domain of the spike, which just means the kind of left-hand side of the spike protein. And so far, I have not heard anybody who has a really strong explanation for what the heck they do biologically, but the virus keeps mutating in that position, and it seems to be in the variants that spread fastest. So, there's a kind of, I think we have to have a kind of humility about, we understand some of these bits and can make predictions when we see a new variant, but actually we also sometimes just get it completely wrong. And then the question about vaccine changing, I mean, for that, it's kind of interesting. I think it almost doesn't matter if we know biologically what's going on. As Catherine said, we kind of put all of our eggs in the anti-spike vaccine basket. All of the currently approved vaccines target spike in one way or another. But they are amazingly easy to swap in and out the code for a new variant of the virus. So I know Pfizer and Moderna, for example, have done, I think, already in human like safety trials of a B1351 version of the vaccine. There's a question about whether we're going to need a booster that's specific to variants or a cocktail of variants or who knows. But it's miraculous, I think, that we talked a little bit about genomics being ready for this pandemic, but the vaccine development platforms, especially these RNA-based ones, are just amazing how easy it is to change exactly what the payload is, and how, in principle, even a variant that gives the vaccines trouble could be targeted by a booster in a matter of months.
Mm-hmm. Yeah. I should just mention, because I see in the questions that there are various questions about variants too. I should mention that another of the panel talks in this in this series is going to talk specifically about variants, and that's the one on Wednesday, the 9th of June. When does a variant become a villain? So you'll learn much more about that there. And with that in mind, I wondered if we could turn a bit to the, I'm interested in the logistics of how COG UK happens. How does it work? How are the samples selected? Out of all those that come in, how are they chosen? How quickly is that done, and how crucial? I mean, I imagine that speed is crucial here to identify new variants. How quickly can it be done? So I don't know, Catherine, maybe this is something for you. How does it work?
So it's changed a lot. It's probably the best way to start. Now we're in the situation that we don't need to select what to sequence, because the case numbers are so, they've dropped, and we've increased capacity for sequencing so much, in the Sanger and in the network, that we have the capacity to sequence everything that's received, as long as there's enough virus there to sequence. So now, like that selection isn't an issue. During the heart of the pandemic, when they were at the peak, we tried to ensure there was random selection of sampling, because I think it's really important, if we want to be able to spot new variants, that you're able to see that randomly in the population, but also ensuring that we capture the critically ill patients in ICU, people in vaccine trials, reinfections. So we had a sampling strategy that we said, at least 50% of our samples should be from a random sampling framework. And then also the other 50% could be used to address those key national core research questions. And in terms of how the samples come, that was the other bit you pointed on. It's definitely not one size fits all. As everyone have seen, the diagnostic testing has changed a lot since the start of last year. So in the NHS diagnostic testing labs, there's different samples that are left after the diagnostic test. They can be either the RNA, which is the easiest sample to sequence as such. they can be live virus, which will then need to be inactivated before sequencing. And then there's just the primary itself. So there's different sample types. So when we get in contact with the diagnostic lab, the questions we have to ask is, how many samples do you have for sequencing? What is the sample type? And depending on the sample type that they have, will determine how we transport that sample. For example, if they're RNA, they need to get shipped on dry ice, which requires a different logistical operation. And then you need to make sure that the dry ice is available, they have the metadata to go with the sample, and that each person from the diagnostic lab and the sequencing lab know when the sample is leaving and arriving. So it's a complex picture, which is why we've had to work a lot on goodwill and really communicating with the NHS sites to send those samples in a way that we could process them. And then of course we've had the issue with point of care testing. So there's no output sample to sequence. So we've been encouraging people to retest those people, so there is a sample for sequencing, so we can spot any new variants. And then there's also issues that we have, as Jeff alluded to, if there's enough viral load. There's some diagnostic tests that there's no quantity of the viral load. So if there's a very low viral load, it's very difficult to get good sequence data from that. So that's called a CT value, a cyclic threshold. So once we know that CT value, we tend to set a threshold of about CT 30. So that would be another part of the sampling strategy that we would try do. So that's the NHS diagnostic testing side. And then in terms of the PETA-2, and Jeff can talk about it more, that the samples are tested at the Lighthouse laboratories, and the output is RNA, and those samples are then sent in RNA plates as daily shipments to the Sanger Institute, and that's a huge operation. So that's kind of point to point from Lighthouse Labs to the Sanger, whereas the other side is a mixture between sample types and how they come, and other logistical challenges is probably the best way to put it.
And are these samples beyond the location of the source? Are they anonymized or not? And if they're not, can people be told?
They're completely anonymized. So when the data comes to the sequencing lab, there's they come with an anonymous identifier that then they get a sequence identifier that gets analyzed. So where the data can be linked is at the public health agency. So they would hold the linkage, and the public health agency then can talk to the health protection teams or the clinical leads to talk about it. But COG UK itself, the sequencing sites, they don't receive any data that's identifiable.
And I think sometimes they do tell people that, when they do, for example, the surge testing that happened for the B1351 variant in London, I think if they get a positive, they phone people up and say, you've got this bad variant, you should in reality, we should probably tell everybody to stay home as best they can, regardless of which variant of the virus they have. But I think they do encourage people even more when they're testing for these particular variants.
Right. Okay. Well, I wonder, we've got a nice lot of questions coming in on the Q&A. I should probably make a start on those. And I'll take it from the top, really, because Eugene Keener, you had your question in first. There's a question here from Eugene about, it's really about the integration of viral DNA or viral RNA into the host genome. It says, is this confirmed, and if so, what are the implications? I suppose that's really a question of whether there is likely to be, long-term.
So there's a paper from some eminent scientists in Boston on this. It is pretty controversial. I would definitely not say that the scientific community accepts this as correct. I'm pretty skeptical about it. So I think it's unlikely, and people will continue to look into it, and I don't think there are really any long-term complications.
And it's important to note that paper that was published through a sort of not full peer review route as well. So it wasn't properly critiqued by the scientific community before it was published.
Mm-hmm.
The Proceedings of the National Academy of Sciences has this unbelievably retro contribution route where a member of the Academy can basically just stick a paper online.
It's led to some really intriguing papers over the last few years.
Nature would never do that, Philip.
Well, I was going to say there is an interesting article in Nature published today by Helen Pearson about this whole issue of how definitions of and the quality of evidence has shifted necessarily, I guess, during the pandemic. So, you know, it's a really interesting issue. What do we take as the threshold of good evidence these days? And, you know, has that had to be lowered or changed, you know, as a result of the situation we're in? So, yeah, I'd recommend people taking a look at that. So there's Linda DuPont asks, how long will we carry on sequencing positive COVID positive samples? I mean, Catherine, you said already we can do them all because there are so few, but you know, clearly we're not out of this yet. Will this continue for the indefinite future?
For the foreseeable future, we will be sequencing every positive SARS-CoV-2 sample that we can, so that what we receive into our sequencing site and that we have enough virus, we will sequence. And that, as Jeff and I have said, we've continued to increase the sequencing capacity throughout the pandemic. So we're no by means downscaling. We're going to keep on increasing that sequencing capacity. So we're ready if the numbers do go up again, or for whatever is next to come in the genomic surveillance, that we're ready. I also see from Linda's question about the most common variant, that would be the B1.1.7, the so-called Kent variant, I think would be fair to say is the most predominant one that's circulating right now.
It is, but watch this space, because it's been almost boring that we've sequenced hundreds of thousands of B117 genomes in the last several months as there've been nothing else, but this B16172 may be beginning to creep up on it.
But we'll be ready, Jeff. We'll be ready.
Yeah, we're ready.
We're ready.
We've just bought some new servers, so like
Verity's ready to write more programs to analyze them. We're all set.
Yeah.
I'm old enough to remember when B117 was anything but boring, I think.
Yeah.
Anyway, so Brian Connellan asks, it's a question actually that I'm interested in about the basic biology of the virus that we've learned about. How mutable is the COVID coronavirus compared to other viral pathogens?
So it's pretty slow, to be honest. So it's an RNA virus, so it means its genome is made of RNA rather than DNA, and that means that it's less stable than a DNA virus. Most of the viruses we care about are RNA viruses, but compared to, I think it's 3 times slower than Ebola mutates, some of that kind of region, and this actually makes our lives quite difficult. So it's good in some ways because it means it's easier to design drugs and vaccines. So like on a sort of applied, like practical level, it is definitely a nice thing. From a phylogenetics point of view, it makes life really hard because so many of the genomes are just completely identical. There's nothing different between them. And everything that, all of the analyses that we run in the Rambo lab are kind of based on these differences between viruses. So it can make making definite conclusions about whether things are outbreaks or how connected patients are to each other quite difficult. We found sequences that are like 80 days apart in sample time and sampled from across the world that are identical. So it does make life more tricky from that point of view. But we do think as well that the other thing that's important with viruses is called recombination, which is where if you have 2 viruses that are sort of genetically a bit different to each other in the same cell, they can do a bit of swapping on their genome. And we know that coronaviruses as a family do this quite a lot. It's been hard to see in SARS-CoV-2 because they're mostly all the same as each other, so it may be happening, but we can't see it. We do now think we found some that have recombined with each other. I think there was a sort of blog post put up about it on a website called Virological.
Rambo Lab managed to find like 6 recombinants out of 1 million sequences.
Yeah, that's Ben Jackson here. Yeah, he's all over this stuff. Yeah, so there are some, but it does seem to be, like not too common, nothing that we need to worry about yet, I think, for that. But that's the kind of the other thing that we think about with viruses.
I think, Phil, I think it's important to say on that note, I think this really highlights the importance of sequencing combined with good epi data. because as Verity said, you can have two sequences, 0 SNPs apart, but you really need that epi data to understand if it's an outbreak or what's happening in the hospitals or the transition dynamics. And we've all envisaged this really good genomic surveillance system where you do genomics and you do epi. But I think we're really now showcasing that you do need good epigenomics to untangle this transmission dynamics.
I wondered, actually, there's a question that might pick up on that about, it comes from, I don't know if it's, Pam, or whether it's, those are initials, PAM, but the question is, Verity talks about the use of data from the Ebola outbreak to work out which are the important bits to collect to create a model for future use. I guess this is sort of touching on that in, how do you, from the data that you get, how do you build any kind of modeling type picture of how things are spreading and how they're related and how the different variants are related, or what is the information that you're really looking for from that database?
Yeah, so I'd say, so the Ebola stuff I was doing is kind of quite different to what I do now for COG, but the Ebola stuff was using, so we can do things like if you take the location of where a sequence was sampled, so that can be like a county or whatever, is usually what we use in the UK, you can treat that as a trait of that sequence. And then you can use the same maths that underlies how you model a sequence mutating over a called a phylogenetic tree, just a family tree of how things are connected to each other. You can use that same maths. So you can look at two sequences at the end of a tree and ask where the kind of node, the branching point between them, where we think that was in space as well as time. So we use that for the Ebola model I was working on, we used that kind of information. So like from the real genomes, and we said, okay, so we know that it jumped between counties in Sierra Leone this many times in this time period. So we can use that number to calibrate our model so that we know that it's got the right level of transmissibility between counties. And that's called fitting a model. And usually you fit epidemiological models using case data and things like that. And one of the things I was trying to see was if we could get this other stream of data using the genomes. What we do with SARS-CoV-2, so like we've got, I'm still planning, I haven't quite gotten around to it yet, but I'm going to do an analysis like that for the UK for the first wave. And we can do things like, instead of saying that sequenced in Hertfordshire, for example, we can say that sequenced at this outer postcode, which is some of the data that we have. So a little bit more zoomed in, but still not identifiable or anything like that, but just more zoomed in. And then we can ask in a kind of continuous way how things are spreading like a wave across the UK. So we use geographical data a lot in these sorts of models. These models are different to what you might have read about in terms of coming out of Imperial. They're more traditional epidemiological models, and they do use genome data, I think, but it's like a slightly different approach, I guess, at least like in terms of what I'm doing.
To maybe pick up on that, there's a question here from Sheila Bird, and Sheila, forgive me, I think I might slightly rephrase this to, I hope I'm going to get the gist of what you're asking about, because it occurs to me then, what is the lag time between getting the genomic data and being able to build a picture of when a new variant is emerging and where it's emerging? How quickly can we do that now? Because clearly that was crucial, just before Christmas. It seemed to take some time before the Kent variant was kind of identified as such and its characteristics were known. Have we got quicker, and how quick does it need to be?
Yes, I think we have got quicker. There's actually two different lags that play a role there. I think one is just what's the time between when a swab is up somebody's nose and when the sequence is in the hands of the analysts churning away at it, and that's got shorter. Back in November, it was probably on average 2 weeks or 3 weeks or something like that, which isn't bad, but it is what it is. Now it's, for the Sanger stuff, it's it's a week basically. It's got we've shaved it down to a week, and I think, Catherine, for some of the other things is even less than that. So that's one lag that we've shortened. The other one is just you see this kind of torrent of data, though, that's flowing across all of these analyst screens, and you're trying to figure out what's interesting and what's not. And I think we have got better at that too. I think all things considered, we were pretty fast, although it would have been nice if we were faster at figuring out that the Kent variant was a real problem. It was spreading probably noticeably in earnest in like late October. I don't know, you know this better than me, Verity, but I think it's late October when you in theory could have seen something, I think.
Yeah, it kind of spotted a bit, and then
and then it was like the first week of December when basically the first warning lights up. So that's 6 weeks or something. It's not anyway, I think it is a lot faster now. I think now we see the data more quickly. We have a sense of what mutations to look for. We can see the growth dynamics changing really fast. So for example, the variants that were seen in India, the B1617 lineages, within a couple of weeks, I think the internal machine of the public health agencies was really already turning and sort of doing additional analyses and putting in place different responses. And so I think now we have a pretty excellent and fast, order weeks system for seeing these things.
I'd say one thing as well that's different now to in kind of November, December is that we're looking for collections of mutations. So kind of at the point where B117 was picked up, we were still thinking in terms really of single mutations. So, last summer, we were worried about D614G, that is just one mutation. And then there was some work done on this one, A222V, which was kind of part of the second wave. So people were concerned that was driving the second wave, when really now we think it was just lots of people coming back from holiday mostly. So it kind of requires a slightly different way of thinking to spot collections of mutations being important. And what was interesting about B117 was that it was a combination, it was like two independent sources of data that was interesting. So it was noticed the kind of clump on the tree with this long branch before it was noticed as, it literally sticks out of the tree. So I was like, oh, that's kind of odd. And then I think Public Health England were like, oh, there's this quite fast growing number of cases in Kent, and that's quite odd. And it was those two streams of data together that suggested sort of independently that B117 was something to worry about. So I guess part of the problem with that is that you had to have the large number of cases and the large number of sequences to notice that there was something going on. But yeah, I do think it would be fast now, because we're like, everybody all around the world is looking for variants. So you get this thing like in the US, there's quite a lot of like, this is a variant. There was a newspaper article that referred to a variant as the devil or something. and then it turned out that it actually wasn't even, it was fine. It's not a variant it's not a variant. It's just another collection of mutations in a lineage somewhere. So everybody's really looking for them now. So I think there is also this like higher awareness of them.
I do think that that comment about collections of mutations touches on something that goes back to what we were saying earlier about the number of genomes that we've looked at. I think we're discovering for the first time, I should say, fundamental aspects of viral biology, which is, probably everyone's conditioned to look at one mutation at a time, that you have a specific antigenic mutation that gets escaped, there's a whole, whereas I think this virus at least is teaching us that it is often combinations, as we were saying, there's a bag of 5 or 6 tricks, and it's playing several of those at the same time that really leads to something interesting. And lots of real world experts weren't looking for that in November, and now they are. So in the space of just months, I think we've kind of learned something about the biology of this virus, and maybe of viruses or pathogens more broadly, that we'll hopefully take into other studying other infectious diseases.
Okay, I'm seeing a couple of questions here about transmission in terms of, we know that there different variants can have different transmissibility, but we know also that there are other factors, social factors, that affect transmission rates, whether it's an urban area compared to a sparsely populated rural area. Socioeconomic circumstances can clearly affect that. The questions also ask about, whether there are ethnicity aspects of that. I'm not aware if that affects transmissibility, but I guess the question really is how easy it is, or how difficult it is, to separate these various influences, biological and social or socioeconomic, in terms of the transmission rates.
Yeah, so you know how I was talking a minute ago about how we can treat a location as a trait of a sequence and then ask how that trait evolves across a tree. What's really cool is that we can also put a probability model on that. So this was a method that was developed in, I think 2014 originally to look at flu, and then it was used to look at Ebola in 2016, and at these kind of big sort of high impact papers. And basically what you do is you give it this probability model, this generalized linear model, and then you can just give it all sorts of extra data. So for example, so like we're trying to do this at the moment, but the variants keep popping up, so we get distracted. We're putting in things like socioeconomic status of an area. We're trying to get proxies like distance to an international airport, things like size of population in the area in the county, all of these sorts of things. And then what you can do with that is it will tell you, the model will tell you how, whether that factor is important for transmission between locations. So you can get an idea of what factors are important for the spread and persistence of a virus. So in the Ebola example, they didn't, there was all this talk at the start about how there are these shared languages across international borders in West Africa, and how the borders are very, very porous in that part of the world. And they found actually that the international borders between Guinea, Liberia, and Sierra Leone did have a protective effect. It did seem to decrease virus transmission between neighboring counties, even though, like, obviously there was some transmission there. But so that's a really important thing to know. So in that case, shutting down borders a bit quicker would have actually been more protective than people initially thought. So yeah, it's those sorts of things we can ask, and we can see whether it is just usually what you expect with these sorts of dynamics is you expect to get big cities have more transmission in them than sparsely populated countryside. That's kind of the basic thing. And the closer you are to a big city, you expect to get more transmission. But that may not be the case, and lockdown affects lots of things. So yeah, so we are trying to run that analysis for the first wave. But it's really not trivial to work with this enormous amount of data, it turns out. We keep having to make new models and buy new equipment. So yeah, it's an ongoing struggle.
It's also, I think, exactly the key fact why, if we want to ask the question, does B1617.2, this new variant that is growing in the UK, is it actually biologically more transmissible? It's not as simple as just to count the number of sequences we get this week and compare it to B117 and say, aha, it's going up or going down. And there's a bunch of reasons for that. One is we got a ton of imports from travelers returning from India. So that kind of, injects a bunch of this variant of the virus into different places in the UK. The restrictions were changing, and they still are changing as we go through this. So it's a kind of, moving thing there. And then finally, the exact communities in which that variant began circulating initially are not a completely random draw in terms of socioeconomic status or of, density of living or any of these factors from the UK. And so you just don't know how to disentangle those things. And in fact, if you looked at the kind of density of B16172 by local authority in England initially, you would see like these massive peaks in Hillingdon and Crawley, but that's where Heathrow and Gatwick are. And so it's, people who are in quarantine facilities getting tested positive initially, and it wasn't because there was some kind of uncontrolled outbreak there. So you just need to be able to try to, feed in these other kinds of data to separate out what is the virus biologically doing from what is the human community in which it's circulating doing.
Right. I guess that also there's a question here about incoming people to the country and whether whether they're if they test positive, whether they're sequenced. I guess, Catherine, you've sort of answered that already, in that everyone who's testing positive, it seems, in the UK is being sequenced at the moment.
Yes,
so
indeed.
presumably we know for anyone who comes into the country, not just whether they have the virus, but which variant they have at the moment.
Yeah.
Yeah. Okay. There was a question also sort of intrigued me as well about, I guess, essentially, it's asking whether the same mutation can occur in more than one place. We hear about the Kent variant. Could the Kent variant arise in India or in Brazil? Have we already seen that, perhaps? The same mutation arising in different places?
Okay, so there's a couple of things there. So the first thing is that the variants that we talk about is not just due to a single mutation, it's due to a whole cluster of them. We call them constellations. So that makes it probabilistically more difficult for it to arise somewhere else, right? Because you're not talking about one thing, you're talking about lots. So for B117, it's actually, there's 23 mutations that define that lineage, and 17 of them actually change the amino acid that it codes for, which is huge. This is why it literally sticks out on the tree, because
and are they all thought to be significant?
No, we don't know what all of them do. Some of them are on that red list. A lot of them we're kind of not really sure. So that's kind of the first thing. The individual mutations absolutely show up a lot of different times across the tree. This is part of the problem that we have with building trees, because there's so little diversity, and then the same mutation pops up in lots of times. So sometimes the algorithm that builds that family tree pulls in sequences to places they shouldn't be. And that's why things like location and time data helps us to build that tree more accurately. But certainly we see N501Y, which is one of the ones that we think we should be worried about. That has popped up in a lot of different places. E484K has popped up in a lot of places. Yeah, so it definitely does arise a lot, which is interesting because it suggests that the virus is always choosing the same selective pathways out of whatever pressure it's under, which is really interesting. From a kind of evolutionary point of view. And it's not clear whether it will then, that's just you kind of think about this landscape in terms of hills and troughs, and it's not clear whether that's just the easiest way to get to the next hill, and there's another hill somewhere else, or whether that's the only hill you're talking about. You know, it may have a limited box of tricks. I suspect it's more likely that this is just the easiest way because it's a virus and viruses are good at adapting, but no, it's only interesting that we see these same ones popping up everywhere. Yeah.
That makes me think also of, you mentioned that this is a relatively slowly mutating virus. If in the future we are faced with a viral pandemic that doesn't have that aspect, where the mutation rate is much higher, will that be could it be significantly more difficult to keep track of variants, to do the kind of work that you're doing?
In some ways it would be easier, I think, actually. We'd certainly be more confident in a lot of our conclusions.
But it could be harder to kind of understand when the virus has got to a kind of biologically interesting place, but because this virus is kind of boring in a way, when, as Barrett alluded to, it suddenly does this kind of, backflip with 20 mutations to a different place. That's so weird that it, it stands out and makes it easy to pay attention to it. Whereas if there were things binging around bit by bit, then you might be less able to see what's interesting. One thing we haven't talked about, I don't think, but which is kind of interesting, is where do those 20 mutations come from? Because usually we see them one by one, happening, and we can sort of watch each one as it arrives. And I don't think we know for sure, but I think there's pretty good evidence that this kind of thing happens when they're infected with the virus for a long time, for like months. So this isn't long COVID where you kind of clear the virus but have after effects. This is just, for whatever reason, instead of having 2 weeks of having the virus in you have it for 3 months or something. And that seems to create because usually the virus has a short time to evolve within one host and then has to jump to the next person. It needs to be ready to go. But this creates an opportunity for the virus to basically swim around and evolve and maybe explore different combinations of mutations that get to one of those hills, those evolutionary hills that Barrett was talking about. And so I think that's been a kind of interesting discovery that seems to have happened several times in the creation of these variants of concern.
Yeah, although we think, so B117, the running theory is that long branch was all in one person. And at the moment, the current estimate in the analysis that we're running, it's about 100 days long, that branch. That's a very long infection.
Sorry, just 100 days before the first sampled case.
Yeah.
Wow, okay.
So
it was cooking early in the whole pandemic then.
Yeah, the most recent common ancestor, which is the node that splits it from the whole background diversity, is depends on what downsample of the data set we use, but it's early last year. It's kind of spring last year. So that's, yeah, really sort of illuminating. We think the P.1, and so the one that was found in Brazil, and the B.1.351, the one that was found in South Africa, we're not so sure that's a chronically infected case. We think that might be more to do with rapid transmission in a kind of very dense community, but it's hard to tell. What's interesting with those two variants, actually, as I said earlier, you know, that, oh, we don't see variants popping up the same all over the place. Actually, those two variants are very similar to each other. They do carry quite a lot of the same mutations, which is why we're viewing them as evolving possibly in a similar mechanism. And then B.1.1.7 is a bit weird and a bit different and has a lot more mutations and slightly different ones. And yeah, I've sort of come to the conclusion, I think the B.1.1.7 origin, I think it must be quite a rare event because there's been so many infections worldwide. And so far, this is the only one we've seen that is quite like that.
Well, escaping is a rare event, but this process happening, there's lots of case reports of like one sick patient who they've studied and taken samples for over months and sequenced them over and over again. And you see sometimes exactly the same mutations, N501Y, et cetera, and definitely this pattern of multiple mutations and multiple spike mutations. So
maybe it's that the selective pressures that make something successful within a host are very rarely good to make it successful between hosts.
They seem to die off basically, sadly, often with the patient where they start, and then B.1.1.7 obviously was something different.
Yeah, so that's the rare bit, that it found the part of the landscape that was really good between hosts as well, because it's the within and between hosts they're almost different fields of study. You know, they're very disparate ways of thinking about things.
There's a question here. Wherever there's big data these days, there is AI, there is machine learning. So the question is, would AI speed analysis in the future? I can imagine machine learning might already be used in this, but if not, would it be useful? I don't know if this, Catherine, if this is something for you to.
Well, I think we can all probably contribute a bit to this. I think, as we said, is sequencing is actually almost the quickest bit. I think is the key bit here. So sequencing can be done in about, a couple of days, even 2 days inside, or even quicker if you want to do it really quick. A big delay is actually getting the sample. So once it's tested in a lab, sent to a sequencing site, and get on the run. So I think AI and machine learning has been used a lot in computational biology. So that is used in different models, and I think Verity and Jeff can expand on a bit more. But what I would say is that if we want to increase or reduce the time it takes, I would say where we really need to target is getting the samples as quick as possible, because I really think that sequencing actually isn't the bottleneck. It's everything kind of upstream of that. I'll pass over to Jeff and Verity to comment a bit on that too, if you want.
Yeah, I can do a quick thing, because I know one example of where we're already using machine learning. So there's a piece of software called Pangolin, which assigns these. So when we've been saying B.1.1.7 and all of this, these are lineages, and it's a lineage naming system that we kind of came up with early last year. But you have to put sequences in those lineages for them to be helpful. And there's this tool Pangolin that does that, which has been developed by other members of the lab group. I think I made a table for it once. It's not really so much what I do, but it's yeah, and in that, so the original model was a phylogenetic model, and very quickly that became, it didn't scale very well with the amount of data. It wasn't very fast and used a lot of memory. So now there's a machine learning approach to assign sequences the correct lineage. So I think it takes a few days, I think, to train the model on. So we give it some data that we say these are right, and it trains on that, and then it yeah, so that's how and there's a web app, so people go onto the web app and they put their sequence in, and then we call it Pangolin runs on it and provides the sequence with a lineage designation.
The one slightly those are both excellent examples, and the one slightly different spin I could put on it is one thing that I think we should aim to do coming out of this pandemic and for the future is to get the most useful data sets possible available to people who develop methods in AI, for example, so they can build better systems for next time to detect a variant of concern the first sequence you get it. You know, people I'm sure would want to try to crack that problem to pull in different types of data. And one thing I'll say is that GIS8, which Verity I think mentioned, is great at basically putting out the sequences and the dates when they were sampled. And then it's a lot patchier with, I think, the next most important piece of information, which is exactly where they were taken from. And I think that's something we could work on as a community for the future, because some places they're pretty good. They'll tell you the country, the state, and the city. The UK, I think, is actually not great at this. We say just basically England, Scotland, Wales, and Northern Ireland, I'm pretty sure. And it would be a lot more useful to at least put a bit more granularity on that. So, you know, AI and those kind of machine learning methods will be great. They need lots of data to work. And so one of the keys will be making sure that data is available to people to build better methods.
Great. Okay, well, just finally and briefly, we just about have time, and I wanted to get this question in at the end. A big report came out today from the, I forget the exact, what it's exactly called, but it's basically the body that the World Health Organization set up to look at pandemic preparedness, and it was quite critical about, certainly about how this pandemic had been prepared for, or had not been prepared for, and how it had been handled. Sadly, we're probably going to face something similar to this in the foreseeable future. Are we now set up in terms of genomics? Do we now have the systems that we need to do what we would need to do in a situation like that? Or if not, what is the one thing each of you would love to see added to that?
I think the system is pretty good now, and I think actually it's a real testament to Sharon Peacock in particular, who's the director of COG, but actually all of the people who were kind of in the room in March of 2020 to say we need to do this for this pandemic in the UK and set up a really amazing system. And I think if I wanted to add one thing to it, I gave in my previous answer, which is just a little bit more of the data attached to these sequences so that you can get as much sort of public analysis as possible.
I agree with Jeff. First of all, I completely agree that, you know, I think Sharon has been instrumental in putting everybody together, you know, getting people from foreign nations and different academic institutions and sequencing sites to all come together to do sequencing as a network and share data between the DAs has been fundamental. And I think, as Jeff said, that data sharing, you know, and the more data that we can get and the quicker that we can get it will be key. And you're talking about preparedness for other pandemics. We don't know what's down the road, of course. If there's a pandemic of a different pathogen, we'll need to learn about that pathogen. But I think we can definitely use the lessons learned from this and the technology and the capacity we've used to kind of to build on that and to be ready. So I think we're in a really good place. And the tech that what we have now can keep building on for the future.
Yeah, definitely. I mean, like it's been a joy to work with so much of the UK data. It's such a wonderful detailed data set. But the only thing I'd say for the UK is it turns out people are really creative with what they think a county is in the UK. So the main, really, it's a very personal gripe. It would be the kinds of things like because all of the kind of interfaces and stuff were set up very, very fast, and that like it was amazing work kind of this time last year, very, very fast, but there were kind of mistakes made in the implementation that made people kind of at the very kind of downstream end of the pipeline their lives a lot harder. But now, of course, like we're not only talking about like the systems that lead us up to the sequence that have been developed now, but we're talking about like a year's worth of really hard work on all of the pipelines that analyze that. So these are run routinely, and they take input sequences and they clean them and they clean the metadata, and everything is done in a standardized way. And the work on making them as efficient as possible, writing new scripts, developing new tools, finding people who know how to do things in different languages that are more efficient than the languages people code in. All of that work has also been done, and that pipeline and that system can also be exported to other parts of the world. Like other people can use that as well, and I think that's a lot of that is the plan. So we're working towards I think Climb, which is the big server, the MRC funded server in Birmingham and Cardiff. I think the plan is to try and set up versions of that in other countries, and certainly there's tools, Civet, we're trying to make that available to, currently you can only use it in the UK for various reasons. We're trying to make that possible for people across the world. So I think part of what will help with pandemic preparedness is taking all of the things that we've learned from a really successful genomic response in the UK and ensuring that other people in other parts of the world can also use it to just kind of help with some data equity and these sorts of issues.
Fantastic. Thank you. Well, that's a very nice positive note to end on. And we do have to wrap it there. Thank you all so much. I have learned so much this evening, and I hope the audience has too. And thank you for everyone who's listened in as well. And now I'm going to hand you back to Ken to hear what else COVID Connections has in store. So thanks very much to all of you.
Oh, thank you, Philip. And yeah, I hope everyone found that as an interesting discussion. And the pandemic is a subject that's affected us all. And I just think hearing what's been possible through the science and in interrogating the genetic code of the virus, is a reason to be hopeful, not just here in the UK, but as we've heard how this can impact, the situation around the world. And it's probably also worth remembering all of this work has happened within the constraints of social distancing. The science community and the research communities have had to follow the same rules as everyone else. So, it really has been a tremendous effort. And as is always the case with these events, there are so many questions, more than we could get through in the time, but hopefully we've managed to cover a lot of the themes that were coming through in the chat. If you'd like to be in the audience for the next event, please remember this is the first of 4 talks, as Phil said. The next being on Wednesday, the 9th of June, when we'll be looking more closely at how the virus changes and asking that question of what exactly makes a variant a concern. And so Dr. Alex Slathbridge will be chairing that event for us on the 9th of June. We are putting links on the screen and perhaps in the chat as well to our events page, so you can find out more about what we're doing at Welcome Connecting Science. And also you can sign up to the newsletter actually if you want to be kept in touch with future events. We'll also put links to the Sanger Institute and the COG UK websites where you can find out more about the genomic surveillance work, and also to Philip's website so you can see more about his latest books and articles. If you'd like to watch this event again or share it with friends, family, then we'll be posting it on our website in a few days' time. Just finally, as we close tonight, we'll post a short survey. Please, if you can, do stay online and fill that out. It really does matter to us what you think about these events. We'll also be emailing it just in case people are short of time this evening. So finally, thank you so much again to Phil, to Catherine, to Verity, and to Jeff for tonight's event, and to everyone out there for joining us. Take care, stay safe, and hope to see you again soon.